Okay.
Ok, ok. Ništa, hoćete da odmah pređemo na engleski pa da onako da uđemo
Može, može.
ok, ok,
Baš
so.
navikli ne znam na hrvatskom. Nisam
Je.
više.
Da, pa nije to uopšte čudno. Mislim, ja slušam dosta slušam naših podcasta, slušam Radio Galaksiju, recimo, i šta ja znam, i gomilu puta kada dođe neki naučnik iz inostranstva, koji je ili završio već tamo, doktorirao i već dugo tamo, i oni ili pređu u toku razgovora spontano, ili jednostavno odmah od početka, znači kažu ja bih da pričam na engleskom, tako je. Ok, mislim, meni neće biti problem ako se negde zabatrgamo, preći ćemo na naš, pa ćemo se vratiti. Okay, so let's start officially, which is not really good move, to start. Let's clap our hands and no, be serious and stuff. Okay, so this is the beginning of our talk. My guest is Professor Dr. Tanja Bosak from MIT. I will say this. I will just say a couple of words, like a keywords, and then I will ask you. I will beg you to do your introduction yourself, because that's where I'm really really bad. So I'm just gonna say MIT,
Yeah.
NASA, and rover Perseverance Mars mission 2020. Is it 2020?
It's 2020, March
Yeah.
20.
Okay. So, hot keywords. And I will also say an article from Nature that I think the last time I checked a couple of days ago, it was top. It was like second most like hottest
Yeah.
article in Nature, and the third hottest, I think, and like all, like in the all publishing, I'm not sure if it's Nature's, like other journals, or even if the set was even wider, but we're talking about something really 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 interesting and talked about. So, Tanja, welcome. Thank you. First of all, thank you for being here, and please introduce yourself.
I'm Tanja Bosak. I'm a geobiologist. I work at MIT. I teach graduate students and undergrads, and I'm a participating scientist on the Perseverance rover mission. So I have a grant from NASA to help the rover find samples of astrobiological value and hopefully collect them for return to Earth from Mars.
Cool.
What I've been
I will
doing
I will I
past
will
5 years.
say, yeah, I will say it's cool, but it's actually much more than that. It's really, really, really awesome. I guess when kids like think about what they wanna become when they grow up, one of the probable like first wishes is something to do with dinosaurs or something to do with space. It's like two top notch. Right. Okay, so I suppose that it came up at your thought about it when you were a kid. I was 100% always thinking about being an astronaut and stuff. Well, if not being astronaut, this is I'm not gonna say it's second best. It's maybe even better, because we were talking about Mars, right? Mars is a little bit further away. It's not really reachable for humans still, right?
No. Yeah.
So we are talking with person who maybe like fulfilled her childhood wishes, or not. Maybe you wanted to be something else.
Be an astronaut.
Okay.
I just wanted to
Okay.
travel. I wanted to be a
Cool.
postman.
Oh, really?
Didn't know about astronauts.
I wanted to be Elvis Presley. So it was impossible from the beginning. Yeah.
Yeah, that's a nice dream.
Yeah. Yeah. Okay. So, Tanya, what brought you to the where we are now? Like Land Rover team and all that stuff, and then gonna go to the main topic of this talk.
Uh, I always was interested in the boundary between living and non-living. So I studied physics as an undergrad in Zagreb, and I missed biology. and always wanted to do something that is not focused on the very small and invisible world, but the interactions among systems. So something that is planet-wide, something that um, processes that really change planetary surfaces. And of course, in Zagreb, I don't think in Yugoslavia there's much planetary science going on. But what bigger process to look at that combines biology and physics than to ask questions about life and its ability to change the atmosphere, to change the surface of our planet. And so when I was applying to graduate schools, I went to Caltech because they were just starting these programs. They've always worked with JPL. They manage JPL, and they have Jet Propulsion Laboratory. That's NASA's main lab that sends all these rovers and missions to space. And I wanted to do planetary science, but then as I joined there, they were just beginning to attract people who would teach and really work on astrobiology and geobiology, and that was it. Once some people like that came, I decided this is what I want to do, and so I switched from planetary science to geobiology because it had biology, but it has a lot of physics and it has a lot of chemistry because it's all about microbial processes, and microbes are the chemists, natural chemists. They can perform all sorts of reactions that we are very boring metabolically, and they are really the oldest forms of life that we think of when we look for life elsewhere in the solar system. So this is what we are this is what we are hoping to maybe look for, the samples from Mars. And that was it. And then I got my job at MIT, and I continued working on geobiology and astrobiology because everything has implications for the search of life, for life elsewhere. Then when the Mars rover mission announced the opportunity for scientists to apply, I put a proposal in saying, I know how to recognize signs of very old early life, and understand what kinds of shapes, what kinds of minerals, what kinds of isotopic signals life can impart on rocks. And since this rover mission was about to start collecting rocks, I said, well, there needs to be a group of people who can really help recognize the best possible samples in which to look for signs of life. So that's how I got on the mission.
So basically, you came from geo. I've heard somebody call you geophysicist, and then you just went to the geobiology, right? And so from very small stuff you went to huge stuff, and then you returned to again small stuff, but a little bit bigger stuff like potential microorganism metabolism stuff like geochemistry. I'm biased because I'm a chemist, and I always look for somebody mentioning chemistry because that's what but whenever they introduce you, they just mention geophysics and then geobiology, and I'm always like, where's the chemistry in that? And the chemistry
physicist, really. I'm really not
no, but
to get the math and physics, but I'm more on the geochemistry side. Geochemistry and geobiology, which is really a lot of chemistry.
Cool. Thank you for this. I'm going to use this as a soundbite, as a little reel. Okay, taken totally out of context, but doesn't matter. Okay, 1 point for chemists. Cool. Okay. Yeah. Yeah. So, of course, that's you cannot miss chemistry. It's like there, right? Okay. So, if I understand correctly, there was a lot of I'm looking of word. I need a word for odgovornost. There was a lot of responsibility, like planning where the rover will end up. I mean, end up like land, not end up. It didn't end up. I guess there was a theme. You were chosen as a member, but there was a lot of people. I guess.
Oh, there are hundreds of people.
Mm-hmm.
There are hundreds of people. A rover is, we are really driving a rover on Mars. So it involves engineering. It's an amazing engineering feat because this rover can even auto navigate. It can drive for maybe 100, 200, 300 meters, even on its own. So it uses some artificial intelligence, and there are people who are needed to design that and build that. There is all this expertise at JPL that at least there used to be to know how to send a robot that will work on Mars, to understand all sorts of temperature constraints, how to power that kind of robot, how to orient the robot, or when it's a good time, how much signal we can get, because there are satellites around Mars that send the signals back and forth, and how to get the bandwidth. So I don't know. And scientists who are on the mission don't know anything about that, and we shouldn't. Whose entire job is that. So, and there are even hundreds of scientists, because you need geologists, you need people who are Mars scientists, you need people, there are even atmospheric scientists, there are people who sent their instruments really for technology demonstration. Moxie, that instrument did its job, and it's now it's been turned off for a year or so, or 2 years. But their demonstration was to show that they can make oxygen out of Martian atmosphere. So there are a lot of different aspects to the mission. So it's not one person or one little team's purview.
Yeah. Were you arguing? I'm not going to say fighting, but were you arguing? Like one team against other? I mean,
no. It's you can't. You can't. There's no single person who has the expertise to understand how to really move this complicated system around. So there are project scientists, and that's a scientist who mostly is in charge of integrating the input from various instruments. What are we looking at? What kinds of rocks are we surrounded by? And how often do we need to sample? There were arguments before the mission starts, because if you think about it, it's billions of dollars that go into missions.
Huge investment. Yeah, huge. Okay, even for Americans, even for you Americans, right?
Oh yeah. That's the price is an issue these days, particularly from our sample return. But you can't afford. Everything needs to be well oiled or run smoothly. And so, there are no arguments once the rover is on Mars. Maybe there will be some scientific disagreements as to whether to collect a sample or not. But there were arguments when the landing site was chosen. And that was before the mission. So the mission itself starts when the rover is launched and lands. And then its job is to really go wherever it finds itself, and then we decide how to, and there are pilots for that. I do not sit in my office. There are really well-trained rover pilots who really understand the terrain well, understand the rover limitations or rover capabilities, so they know, we see the slope, this is not where we want to go, or they can anticipate there's a dune field and we should not go that way. So it's in their hands.
Yeah.
But the landing site workshops, that is a couple of meetings that the entire community is invited to. So whoever wants to can come to those, and people present cases for different landing sites, and that can turn into a bloodbath.
Cool.
And ultimately, it's not really the people in the room who choose. We voted. I went to, I think, 1 or 2 of those, and there were some really strong feelings in the room. But the community voted after all these presentations where people argue, we should go here because of these reasons, and we should go there because of those reasons. So we voted on what's the best, geologically most interesting place, and what's the most astrobiologically interesting place. And then it was the then NASA administrator who took that into consideration, and then he made the decision where to go.
Okay.
So
Okay.
that's
So
where the
you're
argument stop.
Yeah. So you're are you suggesting that there was a lot like an alternative landing site? So it could happen that could have happened that rover didn't land on near Jezero. I'm gonna call it Jezero, not Jezero, but okay.
The mission called to Jezero. I tried. I was sad about that.
Yeah. It was
It was too late.
yeah.
Yeah, there were multiple sites. There was anyone, the community, people from the community, the Mars scientists, or anyone really, was invited to make a case. And so people look at the Mars geology and they try to understand. Of course, there is the element of novelty that plays a role. It would be really hard to argue. People have argued for return to some of the previous sites. What was it, Columbia Hills? But it's very hard to go against the novelty of a different site, because there's only
Of course.
rovers that ever go to Mars. And
Yeah, of course. It's yeah. Do you have somebody in your team who's like responsible is sending mails to those people and just with attached couple of these new publications, and just to say, I told you so. You were wrong. That's a suggestion.
No. Okay.
We say hi to those people. Okay.
Yeah, people come and then it becomes a scientific argument. So people present the case for a site. Here is what we know about the geology. Here is what we could learn here. And then, of course, different people, depending on what they study, have different priorities. So they may feel strongly about one site.
Yeah. But geo, and then something else. People are probably the ones that should be asked first, right? Because, we are talking about like a planetary investigation. Like it's geo people. It's like a huge. Yeah. Okay, including yourself, of course. Okay, so could we talk a little bit about those reasons, those particular reasons that were key reasons for choosing? Did Perseverance land on Jezero or near Jezero?
It landed in Jezero. So
in Jezero, cool.
Yeah, it's much easier to land the lower the point. The landing site, topographically, the easier it is to land because it has some atmosphere to help it break. And it landed in the crater proper. So it landed, the goal was to land maybe on that faint deposit. There's that apron-like deposit that told us even from the orbit that some water carried sediments, and there are sediments in the crater right next to the channel that was carved out by some river or flow. And the idea was, or the original goal was, to land on that vein, but we landed actually a little bit further off on the crater floor.
So basically, before Perseverance landed, when you were discussing about possible landing sites, it was already like I'm going to use a known fact, but maybe you can correct. Maybe you can give me your input, but there was like a known fact that there was sometimes water on Mars. That was a given, and also that Jezero was actually a lake, or could we call it lake? Okay, but why was it interesting, like geochemical, biologically? Why was it interesting? Why did you want to go particularly to that place on Mars?
We wanted to go. All these missions have, well, this is why we are talking today too. There's this astrobiological potential. that sparks curiosity in people to look for old life or to understand how life began. So we wanted to go to a place that was a big factor that played into the selection of the landing site. Something that would have been habitable, meaning had standing water, and something that is old. And Jezero is, it's probably 3.8 billion. The crater itself is thought to be maybe 3.8 billion years old. And we saw from the orbit, this is much before the mission. There are orbital maps of minerals in the area that showed us that there are some minerals that even contain structural water. There are clay minerals. So all these minerals that water makes.
Okay, so when we say, I need to just interrupt you. So when we say structural water, we actually mean like water inside of crystal, right?
Exactly.
Okay, so it's like times n waters, molecules of water, right? And if the crystal has a water, I guess it changes its features or like color or something, so you can determine, oh, this one is hydrated, this one is not, right?
Yeah, the hydration, sometimes it's just the hydroxyl group, OH.
Okay.
The previous water. And you can detect that in infrared spectrum of the minerals that can have quite strong features. And that's detectable from the orbit. So there's some orbit around Mars, and it would map the spectra of different regions. And some of those signals were detected. And also what was really intriguing about this particular site was a strong signal of carbonate minerals. And carbonate minerals, just to, I know you know, but these are the minerals similar to what limestone on Earth is, vapnanates.
Okay.
These are minerals that precipitate from
Vapnanates, okay. Yes.
Precipitate because ions in a solution react together, and these ions are cations that are calcium, magnesium. On Earth, it's mostly calcium. And they also combine with carbonate, which is usually in equilibrium with, on surface with atmospheric carbon, so CO2. So we have this record of old carbon. And Mars actually does not have a lot of places with carbonates, unlike Earth. And one of the major questions is why and what kind of climate it could have had to support the formation of some of these carbonates, and why are carbonates so intense in this particular place. And also where carbonates precipitate, we know that the pH is not very high, it's not very too low. So talking about habitability, we know a system that is similar to what we would expect from a habitable environment.
Like Goldilocks zone. Yeah. Okay, so I watched your lecture, this year's lecture actually, and I think it was like Goldschmidt something Okay, yeah, and it was titled, I think carbonates were in the title, so somebody would expect, reading the article we're going to talk about, somebody would expect you will mention like Gregit, whatever its name, Vivianit, like those like those hot keywords. But you talked about, you talked about them too, but you chose carbonates to title your lecture, which was interesting to me, but like you said, so carbonate.
Before the paper was published.
Okay. Was published. Was published. Yeah. But you didn't. I think you didn't even give a hint. I mean, now with no. Yeah. So you were like threatened.
No, we cannot advertise, or we cannot really discuss data that are about to be published.
I'm gonna watch that video again. I'm gonna look for like a body, some like Twitch or something. Maybe. Okay, but I guess that was really hard because that was it's a huge thing. I mean, not only huge potential thing. We're going to talk about null hypothesis and stuff. So carbonates were interesting because they were not. They're rare at the surface of Mars.
Yeah.
I want.
Not on Earth. On Earth,
Yeah, yeah. Yeah. So I want to ask you just one question that's related to that lecture. When you mentioned scenarios for abiogenesis, you mentioned like two camps of ways of thinking. Two ways of thinking. One of them was related to deep water hydrothermal vents, right? And then you mentioned the other one that's more like surfacey chemistry stuff, right? And then you said I want to talk about these surface phenomena, right? Why?
Why? Because here's why. Scientifically, the experimental work that has gone into understanding chemical organic synthesis under surface conditions, even on early planets, is much more developed, and so that scenario has a much stronger backing. When you
really
try
cool.
to look for experiments that make something under hydrothermal vent conditions, I have not found much. You can make some small, maybe if you have the right metal here, it invokes a lot of special conditions, and it doesn't make much that the life actually needs to start. Okay, so you can make small organic acids, like maybe acetate, that really horrendous yields. The yields, when you look at those papers, hydrothermal, I don't know, production of pyruvate or something, the yields are horrendous.
Okay.
Doesn't make nucleobases this kind of chemistry, or at least it hasn't been shown. It's hard to, I haven't seen anything convincing about lipids. Maybe there's some Fischer-Tropsch, but then we are talking about conditions that are hundreds of degrees Celsius. And so in terms of an integrative chemistry that can produce a whole slew of molecules that then can go on to combine into something prebiotic, surface conditions seem to work. At least there's an experimental demonstration for that.
Yeah. That's really awesome insight, because I wasn't aware of that. I mean, that's not my field of interest. I'm doing something else, but like I'm a hobbyist and I like to read stuff, and just to try to put it in a perspective. So you're saying that experimentally, that surface like scenario of abiochemistry, abiogenesis is like not more plausible, but more confirmed experimentally, right?
Yes. Yes. Of course, the science of the origins research, and especially the experimental part, it's only beginning.
Yeah, of course.
So there's not much in either camp, but
it's really
more in the surface camp.
Yeah, it's really a young field of research, I would say. I mean, it's like a couple of decades old, right? I mean, thought about it before, but something that can be done in the lab, it's relatively new. And the articles you referred to when showing those slides, one of them was like 2020. So it's like
exactly.
really. Yeah.
Exactly.
What is interesting is that if we try to put like a pinpoint one of the major differences between these two scenarios. I mean they are different, very different, but one of them would be some kind of energy source, I guess. Surface abiogenesis would probably be open to the UV light and the stuff, but the hydrothermal vent scenario would have some kind of heat as a source, and you need some kind of energy, right, input to like push the reactions. We're going to talk about some reactions. Okay, cool. So let's get back to our.
And I don't think the two are mutually exclusive. You could imagine having some hot spring at the surface. Some of the reactions really do need UV light, or they need freezing or some kind of evaporation to concentrate different reactants. And yeah, they do. Some of these reactions do rely on UV, something to destroy the molecules that could build up otherwise and make a mush instead of a specific set of products. But there's nothing to say that you can have a hydrothermal system close to the surface and then combine it. So you that could work together.
Just one small question. Does Mars have hydrothermal like or X or like X hydrothermal? I mean, not. There's no. I guess there's no liquid water right now at on the surface, right? But did it have hydrothermal? Ah, okay.
Well, here's the. Of course, that was. Well, I said I prefer the surface because we are in this former lake. We have been. It's also a lake that was in a very active, impact modified terrain.
Okay. Yeah, crater, right? Crater. Yeah.
And there can be hydrothermal flow that's induced by impacts. It could be just local volcanism. And so we've always been keeping an eye for something that could tell us, oh, yeah, there was hydrothermal activity. And those systems are typically very local. And so it's hard to find evidence for that. When you have a little robot that can only make 100 meters at most per day or 200, if it just does nothing other than drive. And so it's hard to recognize that. So I don't think at this point we have very strong evidence for, at least in Jezero, for that kind of system. There's every reason to expect it.
Yeah. Just another small question. You mentioned that there is some kind of AI involved in the like programming of rover of Perseverance. Does Perseverance update sometimes? Does it have some kind of really cool?
Yeah.
So I guess that now it's like smarter. I'm gonna use that word.
Yeah.
That's awesome.
Sometimes you start and sometimes there are bugs that or some little glitches that are missed. Of course, these missions are highly vetted, but there can always be some little glitch in some part of the software for some instrument, and they can send a patch and they can
patch. Yeah. Cool.
So yeah.
Cool. Okay, so we're now going back to Mars, and Perseverance landed the way you want it, so or almost the way you want it, right? Yeah. At the bottom, right? At the bottom of the lake, X Lake, or whatever.
At the bottom of on
crater. Yeah. First it was a crater, and then it was filled with water, right?
Yes, at some point. And that's another question that we had. We knew that there had been water at some point, but the question is, for how long? Was it something that was 1,000 years and then it was over? And when exactly in the history? Because we have 3.8 billion years to account for.
Yeah, when you mention 3.8 billion years, actually that's how life, that's how old life is, more or less. Like take give or take a couple of hundreds of years on Earth, right? So it's like there's a interesting parallel, right? And I think that the oldest rock on Earth, like the oldest visible like land, I think it's somewhere around that kind of that age, maybe a little bit younger.
There are some metamorphosed rocks in Greenland. There are some rocks that are highly metamorphosed, but they're igneous. There are some rocks in Canada. I think mostly in Canada.
Yeah. So we'll just explain igneous. When you say igneous, you mean what you mean by like okay. Maybe, so so they're
magma at some point.
So they were melted, right? And then okay. And there is other type of rocks or other types, right? Something that maybe you aimed for, right? And that other type is
sedimentary. We want
sedimentary. Okay. Why? Why do we want sedimentary?
Because sedimentary rocks can form in the presence of water.
Okay.
That's what we find on any beach. If you go to the beach in the summer, you're surrounded by sedimentary rock.
Okay, so sedimentary actually like means like points to the water. Sometimes there was a water, right? There was a solution. There was a solution of something, and then water went away. Okay, and then that solution, that those particles, ions, whatever, crystallized, right, and made okay. So that's interesting. So sedimentary means sometimes it was water. Cool. Okay, so
wind. You could have old wind formed dunes that also become rocks, and so you can see them. So it doesn't have to be water. That's
okay.
Questions we have when we see something that looks like a sedimentary rock. How is this visited?
But based on the looks of that place, Jezero crater, you were pretty certain that there was sometimes water because it had kind of rivers, right, leading into it, or maybe out of it too. Both, okay. So there was
that led in, and there was a little outflow channel at one point. The water level must have been high enough to break the rim of the crater, which is slightly lower on that side, and higher. And then the water flowed out.
Okay. So first goal achieved, right? Perseverance lands on the crater, old sedimentary rock. And did it start taking samples right away, or did it need to go somewhere to? I mean, there were like 30. There are like 30 samples, right?
Yeah.
So did it start taking samples right away?
No. It's like every you buy I don't know, a sound system, and you have to start turning on the buttons and connecting them. And once we landed, the whole team, the science team included, was on this Mars time for, I think it was maybe 3 months. It took a while for all the instruments, because there are a lot of instruments, there are a lot of cameras, there are a lot of these chemical analyzers on over. And so each instrument had to turn on and make sure it works. Okay, so everything has a system and safety procedures and a series of analysis to make sure that things work. So that's in time.
Okay, so did you already have like coordinates of some potentially interesting places to drill, or did it depend on what the Perseverance will see, like quote unquote see?
We had some idea, guided by the observations from the orbit. So we wanted to sample some of that fan where there are sedimentary rocks. We didn't even know what the crater floor was made of. We were hoping for very, very fine-grained sedimentary rocks.
Did you get it?
Nope.
Okay.
It turned out to be volcanic.
That
was a surprise. And we hope to get well, of course, you can always put a wish list together. And so we had a wish list, maybe some rocks that were made in hydrothermal environments, and some carbonates, so some rocks from the area that has a very strong carbonate signal. There are, of course, always samples of opportunity. We also budgeted for a sample of soil, regolith. And but everything else was really open to what we saw as we moved, and that's also determined by where we can actually go, how much time it would take, and then the team does make some decisions on where do we go, and argue. There can be some, constructive. Back and forth.
Yeah. I know all about constructive talk. Okay, so but eventually. The rover started drilling, right? Taking some spectra. You were talking about payload, Perseverance's payload. You said a lot of expensive cameras and instruments. I guess I'm not guessing. I watched your lecture. So IR spectra, right? Probably UV or like X-rays and stuff.
It doesn't have an X-ray.
Okay.
No, actually it does. It has an XRF mapper, pixel. That's the instrument that makes these maps of maybe 120 micrometer spots. So you can profile the chemistry of a patch on a rock.
Yeah, we'll come to the patches very soon. Okay, so and it starts recording, sampling, and stuff. What was the first sample that got you excited? I guess the first one. Was because it's the first one. Everything works and stuff, right? But was there some exciting sample before the sample number 25? We're gonna move to sample 25.
To me, all the samples of sedimentary rocks. So the very first sample we tried to take fell apart.
Oh.
We decided it was an igneous rock, so this volcanic rock that that had some indication of having seen water. So some igneous minerals that are produced in the magma when they cool and you get a rock. Sometimes water can interact, just flow through the system and change some of the minerals. Well, we drilled the sample and we have these cameras that can look into a tube to see that the sample is there. And there was nothing. And then it took a while. Is there something wrong? Is the drill okay? Is everything
So the
working?
first one was like a problematic one, right?
The first one, there was no sample. And it fell apart. It turns out it must have shattered. It must have been so eroded by water that we couldn't collect a whole solid core. And so we just capped the tube and called it an atmospheric sample.
Oh, hey, that's what I wanted to ask you. So does it have a number? Is it 0?
Yeah, it has a name. Every sample has a name.
Yeah. Okay. Yeah. So it does have it does have its place in history. Let's just say that way. Okay. So you said every sample that was sedimentary that took sedimentary rock was interesting for you, right?
For me, yes. For me, it's all these carbonate rich samples. So most samples from that vein, although we did not think the carbonates would be so abundant in the vein itself, maybe a little further off, close to the rim of the crater would be where we expected a lot. But we ended up finding a lot of carbonates all throughout the vein.
Is it because of the contact with the atmosphere, or I guess because of the carbon dioxide from the atmosphere, or something else?
Well, that's probably the case. That's why it's exciting. I find it exciting because that would be the closest to the atmosphere minerals that we collect. And so by analyzing those types of minerals, we can start understanding the past atmosphere, and we can understand how the carbon and the water cycled at the surface in these habitable environments. So to me, that was really cool. Also, by getting it's not just one sample. The whole point is to collect a series of samples in geologic context. We know where they come from exactly. We know what's up, what's down. So we know what's older, what's younger. And so we can get a progression, and we can understand processes in time and the deposition of these types of rocks and the water.
I understand.
And so to me, that is really one of the greatest, the potential to really reconstruct these types of processes is something very exciting.
So now the samples are taken, and it goes. It was the frequency of taking samples is, I guess, I don't know. I mean, if you took 30 samples, though, that means I don't know. But maybe that also changed, right? Depending on the surface, the terrain. Okay. So should we go to the sample 25?
Sure.
Okay, because that was the hot stuff. Yeah. Okay, and then there comes a day when it was drilled.
I don't remember.
Okay.
I don't remember particular.
I'm going to ask you. Okay, I'm going to ask you one thing. So the sample is taken. The drill is doing its job, and
then there you said there are like you have a little photograph or whatever like of the sample, and then the information comes. It takes, I guess there's some lag time, whatever you said. The satellites are the problem. I mean, they should be aligned and whatever, but eventually you get the information. Do you have some kind of alarm system that goes like ringing, oh my god, this is really interesting, you should need like a I don't know, like a AI powered
No,
oh, this is no, okay, so
no,
people.
no, no. No, the way the decision about a sample is made is because we already have used all the instruments and
Okay.
have seen what the rocks are like.
Okay, so
And we don't learn about Mars by collecting samples. We only have
okay.
30, 42 tubes altogether. So,
Okay. So you were already aware of those leopard, rather that they're called
yeah,
leopard spots
yeah,
and poppy seed like
yeah,
points. Okay,
yeah.
so that was before the sample was taken. Okay, cool. But when you saw those spots, what was the reaction?
You know, like every
Okay,
team, different
okay,
reactions.
okay.
I think it was certainly the most interesting and
Mm-hmm,
intriguing, visually intriguing sample that we had seen throughout the
mm-hmm,
mission.
mm-hmm,
Or at least it had some features.
mm-hmm,
Otherwise, it was all these igneous rocks and it was
mm-hmm.
grains, but you didn't see, there was nothing to see in terms of, um, well, anything to even give a name, like all leopard spots, poppy seeds.
Yeah, 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 yeah.
And so that was intriguing. And there, of course, people tend to, it's interesting, for me, it's interesting to
Mm-hmm,
watch um, how people react. Some people
mm-hmm.
will all immediately go to biology and,
Okay,
oh, some biology can make this, and
okay, okay.
therefore it has to be biology. Or
Yeah, yeah. Of
then
course. Yeah.
then the argument is, and some people will be, well, I don't know, this looks interesting to me personally because of what I study.
Mm-hmm.
Anything round is not a hard shape to make abiotically.
Mm-hmm. Okay, okay.
Fusion
Understand.
will do that for you. And
Okay.
so, but I still do think, and here's another interesting and really unique aspect of this sample. We had been looking for organic matter throughout the mission.
Mm. But we forgot to mention. Okay, cool. Okay, yeah, yeah, yeah.
Yeah, so we do have Raman spectrometers, two
Mm-hmm.
different ones on the mission. And there have been some claims of some, there was some also, what is it, luminescence detector
Mm-hmm.
that was designed with the hope of detecting organic matter.
Mm-hmm. Mm-hmm.
Turns out it's a little, it's not the best instrument. It's susceptible to a lot of interference from
Okay.
inorganic things that can make similar
Mm-hmm.
signals.
Mm-hmm.
And so we've been looking because organic matter, you need organic matter to start
Of course.
life. And of course, everything is much stronger as a biosignature if you see organic
Of
matter
course.
minerals. And we had not really seen anything too convincing throughout the
Mm.
mission. And then we
Mm-hmm.
came to this area, and there was a detection that seemed to be above the noise level of something that is organic, this
Mm.
macromolecular complex organic matter. And so
Mm-hmm.
that is also what
I understand.
inspired
We can
inspired the
say it
collection.
was, yeah, because we can say it was one of the I'm gonna call it green flags or whatever. It was like,
Yeah, yeah,
oh,
yeah.
that's something interesting. Yeah, yeah, yeah. Okay.
That's something
Eh,
interesting. Yeah.
yeah, yeah. And
And
then
so
yeah, yeah, yeah.
yeah, that's why we collected a sample. It was
Mm-hmm.
really the availability of all these pieces of information.
Mm-hmm.
It had organic matter. It had these weird features. It had these
Mm-hmm.
interesting minerals that would be made by biological processes. And
Mm-hmm.
the idea is always to get the sample and hopefully bring it to Earth. So
Yeah, yeah.
there are many, many labs and scientists who can really pick the sample apart and
Mm-hmm.
attack it with
I understand.
various techniques and
Of
methods.
course. Yeah. So the sample was taken. The spectra were done before, I guess, and solved, or at least there's a strong indication, right, that there are some things. They're organics. You said it was maybe not the best instrument, but still you have a solid case that it's organics, right? They have characteristic peaks, I guess, or whatever. So okay, and back to the leopard spot and poppy seed point. So you figured out that there was those interesting minerals. I mean, did you know they were there before, or that was also something that was sampled in the sample 25? I'm just calling it sample 25 because I cannot remember its name. It's like a sapphire
canyon. Yeah, sapphire canyon.
Yeah, okay. I'm gonna call it 25. It's easier to remember. So yeah. So were you aware? Okay.
Yeah, the same instruments what we do is for any sample, we abrade a little patch to understand the chemistry of the rock. And so there's this, I think it's 5 centimeters wide area of the rock just to remove the surface and remove anything, any kind of salt or crust that would be on the surface, to remove the dust. And so these instruments have a very flat area that they can analyze. Okay. And so Pixel, this X-ray fluorescence spectrometer, and the Raman spectrometer, Sherlock, and other instruments analyzed this abraded patch and even areas with some poppy seeds and leopard spots, and saw these variations and the presence of iron sulfide minerals, so greigite, and iron phosphate minerals, so vivianite, and some gradients.
Why are they interesting? Why are they as you said in the publication, potential biosignatures in the context of them being related to and near organic compound? No problem. We can always cut this part. No problem.
Okay.
Opušteno. Totalno opušteno. Kaži ako možeš da nastaviš.
Ne, dobro, mislim da je otišao.
Sehr cool. So, why are those minerals interesting in the context of potential biosignature stuff?
These minerals are also, we've been looking for rocks that would have elements in different redox states. So iron that is oxidized and reduced, or sulfur that is oxidized and reduced, because on Earth that is typically, you go to sediments and you'll see some rusty minerals, and then you will see that microbes, when they have organic matter, can use them to respire, so they'll breed rust instead of oxygen, and in doing so they will produce reduced forms of these elements, sulfur, iron, and these were also the first rocks where we saw really good evidence for a lot of these different oxidized and reduced forms of iron and sulfur. And so the spatial distribution. So those leopard spots, they're round, and those rims seem to contain more reduced sulfur and iron.
I was watching a clip by Neil deGrasse Tyson, and he said, you said they breed out. He used the word microbe poop, but I guess that was a different thing. That was just for the audience, just to have a laugh. But actually, it's more like breeding, because breeding is like taking electrons and dumping them, right? So it was like it's electrons. It's redox. Life is redox system.
It's all about little currents. It's all about electrons moving from one compound to the other. They extract, and we do the same. We extract electrons from sugars and dump them onto oxygen.
Yeah, and also we yes, true. Yeah. And also we breathe out the oxidized carbon dioxide, whatever. So it's like redox.
It's redox.
I need to quote said Georgie. I do it. I did it a couple of times. He said life is nothing but an electron looking for a place to rest.
Yes, that's it.
Yeah. That's totally like poetically, but it's really what life is. So by finding these, I'm gonna don't want to make a mistake. Reduced things like iron and sulfur, right?
Yeah.
You were able to, in the context of organics, you were able to suggest that it could be the traces of something that looks like life. So you basically found some electron dumps, right?
Electron dumps, localized dumps of electrons. Yes.
Okay.
And that is what you see. There is another analogy. You can see some spots that look like that, maybe not exactly the same minerals, but in modern day river beds or really on river banks where you have a blade of grass. So you have organic matter.
Yeah.
And then so these are a little bigger. And then because there's this organic matter and there's a lot of rusty iron, this oxidized iron, microbes around will take the organic matter and breed this iron, just dump the electrons and make these action spots.
Yeah.
So
I'm going to ask you something that everybody asks you, I guess, but we need to state that are there some abiotic mechanisms to get there? I know what you're going to answer, but I want our audience to listen to hear. Yeah. So are there some inorganic reactions that can lead to this type of pattern or whatever? Okay.
Yes.
Okay.
There are, and that's something that we don't really have the capability. This is why this is a sample return mission. We have our instruments are good for what we are doing. You cannot use them to really start addressing the potential for all these abiotic type mechanisms to create similar features. And so, yes, you could imagine, I've heard everything when we were presenting these results. You can have high temperature processes that will reduce sulfur, sulfate. You can have certainly iron, they will do that the same. And as I said earlier, little dots of reduced minerals and even round-ish features are not difficult to make abiotically. That's really, if you imagine sediments where nothing is moving and you have some reduction by whatever mechanism you want, you likely get something that looks circular.
Yeah.
And I've heard even some hypothesis, well, maybe there was some meteorite that would contain organic matter and nickel and zinc, all these enrichments in elements that are redetected in the same area. And that could cause local reduction or mixing in of that material. So there are a lot of hypotheses that would need to be tested before we really claim, oh yeah, this is life.
But one particular thing that I think I read it in that article, because you dedicated some space to the null hypothesis, like what could be
not too much space.
Okay, what would be null hypothesis in science?
Yeah, so null hypothesis in science, or when I'll make it more specific. When you look for life or evidence of past life. The null hypothesis is that it wasn't, especially so on Earth, that's easy. You can assume pretty safely, unless you're looking at really old rocks, that everything was colonized and infested
touched by life. Okay, yeah.
Touched by life.
I'm going to say touched, but infested. Yeah, it's totally infested. Okay word, yeah.
It's hard to find something that hasn't been. Yeah. But when you go to Mars and you're looking at 3.8 billion or 3.5, or who knows how old these rocks are, you can't assume automatically that whatever you see and whatever looks weird is life. And so the null hypothesis is it was some abiotic mechanism that made it. Only if we keep making measurements and keep finding additional pieces of evidence that really challenge this abiotic null hypothesis, whether it be a meteorite, whether it be something thermal, maybe there was some metamorphic event. I mean, it is an impact crater, and there have been impacts all around.
Yeah.
And only if we keep seeing and producing new analysis and signals that just can't be explained by those types of processes, then you start building the case for this must have been something biological, simply because our abiotic hypotheses do not work.
Of course.
So that hypothesis.
Yeah, I'm gonna just mention one of the points in the null hypothesis. You mentioned higher temperature, but then I think in the same article, I think it was stated that so far that scenario of higher temperatures in that place are not really not, I'm not going to say possible, but there's a lower possibility of something like that, higher temperatures.
Now that is the point where one really needs to look for. So you would have to look for minerals. You would have to make experiments. So this is really, I think it's a fruitful area for anyone who knows how to look for high temperature phases and predict, okay, if we have this assemblage of minerals, these different types of minerals, and they were subject to higher temperature fluids, here's what I would expect to be produced. And do some modeling and then see, because we're also limited by the time we have at any particular site. So we did not go and spend a month there and even look for those types of minerals. So I would say it's easy to write a sentence where we say, oh, this is more plausible or not.
Yeah.
But it's much harder to justify that. And I think the sample is interesting enough. It's certainly the most interesting sample we've collected, as you
And I would just say, I would just venture, even if some future evidence confirms the null hypothesis, I think that even abiotic scenario is also interesting, because it may hint of maybe something that was happening before life, right? Yeah.
Or it tells us about how a dead planet without really vibrant life, receives organic matter.
Of course.
Why is that organic matter there?
Yeah,
we know there's tons of it's possible to get it by meteorites, from asteroids. But how frequent was that? What's the budget of that kind of material on an early planet billions of years ago? So I think it's a fascinating sample no matter what.
Yeah. Even if null hypothesis turns to be the truth. Okay, so we're gonna go to the next topic, and I'm not gonna bother you much more. You already mentioned that return mission, Mars return mission, or something.
MSR,
MSR,
Mars
yeah. I guess that's something you think about from time to time.
From time to time. When I applied to be on Mars 2020, everyone was excited. It was really probably the most exciting, typically the most exciting prospect. The return of Mars samples. This would be something bigger even than the Apollo samples that are still feeding a lot.
Yeah. Weird.
You're getting these samples from Mars, and here we are studying processes on Earth and saying, well, this could have implications. And here's now we would have to put our mind where our mouths are, really get into the samples that are actually from Mars. Can you imagine?
Yeah. And they're sterile, or I guess we had meteorites and stuff, but I can only imagine what procedures would have to exist, or they even already exist, to keep those samples like sterile or whatever, like no touching, like robots doing stuff. I'm imagining a lot of SF, but maybe I'm not that much far from truth, right?
Yeah, there are working groups. So this is
Mm-hmm.
not part of our mission. We are just
Okay.
collecting. So we are the first
Mm-hmm.
leg of this Mars sample
Mm-hmm.
return, and our job is to collect the samples, the best,
Mm-hmm.
the scientifically most valuable samples we can find, based on
Mm-hmm.
what we see at this landing site. So I think we've done a pretty good job.
I want to say job done, job well done.
Job done. Yeah. Um,
Yeah.
and then the subsequent legs, there's the idea was to really follow. It's like the movie Martian, The
Yeah,
Martian.
okay. Yeah.
It's based on a real plan to bring missions back from Mars because
Mm-hmm.
it's like every planetary mission and anything in space exploration, launching things and
Mm-hmm.
certainly launching things off planets is prohibitively expensive. You
Okay.
have to bring so much
Mm-hmm.
fuel, not only what you need to land, but then you would have to bring a ton of fuel, which is really just gets incredibly expensive. So the idea was to split it in three legs.
Mm-hmm.
And one leg would be just our rover. It lands, it has no capacity to lift itself off the planet.
Mm-hmm.
It
Mm-hmm.
collects the samples in tubes, it caps the tubes so they're sealed.
Mm-hmm.
And these tubes can stay on the surface of Mars. They're currently in the belly of the rover. There's another cache
Mm-hmm.
that has been deposited, maybe 10 tubes or so at the front of that fan. And the second leg would be another little rover that would get the samples from our rover
Mm-hmm.
and have also this Mars autonomous vehicle, and it would be able to lift the samples into the orbit around Mars. So it's
Mm-hmm.
really like The Martian, the movie.
Mm-hmm. Yeah,
Remember
yeah,
how
yeah.
he was, I'm assuming you've seen it.
Yeah, yeah, of course. Yeah, yeah, yeah.
Yeah, he escapes and then is in the orbit, and
Yeah, yeah.
then that orbiter is intercepted by another vehicle because it's really even more expensive to get something out of the orbit and go
Mm-hmm.
towards Earth. So the part of launching to the orbit is not as expensive on its own as
Mm-hmm.
doing everything at once. And then there's an intercepting spacecraft that would get the samples and bring them to
Mm.
Earth.
Yeah.
And
To
so that
America,
was,
I guess, right?
sorry, to
To
America. Oh
America,
well,
of course.
of
Of
course.
course. No, 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 of course. I was watching that press conference 3 months ago when the chief guy from NASA he was, I guess he's a politician, and so he was very like, yeah, he was very politician-like, and it
And
was
that's
like we are planning to place American boot on Mars and that stuff. Okay, yeah, yeah, but of course, I mean, it's NASA mission, right? So you were part of the first leg, okay? But does that mean that you won't like analyze those samples, like you and your team?
no, no.
Oh, okay,
We
okay,
were just in charge. No, the
cool.
idea is to bring these samples back, and then they would be at the disposal of the entire scientific
Oh,
community.
okay.
Anyone with an idea. This is how people apply these days
Okay.
for Apollo samples.
Mm,
If you have an idea and you can convince people that you have the right facility to
mhm.
analyze whatever you want to analyze,
Yeah.
then you write a proposal and you submit it, and there would be a central repository of
Mhm.
samples where they would be stored.
Awesome.
Maybe NASA Johnson, maybe some other place,
Mhm.
very sterilely under conditions that don't damage. They keep them cool. They don't irradiate them with X-rays,
Mm,
and you could apply if you have some
mhm.
chemistry to investigate.
Yeah.
I need 3 grams of this sample. That would be a lot, actually. But
Yeah, yeah.
and
You
then
mentioned. Mhm.
if your idea is good enough, and there will be certainly a board, some kind of
Mhm.
scientific board that would decide
Mhm.
this is a good idea, and we can dedicate this much sample. Is it a destructive analysis or not? And the idea is for these samples to fuel science, to stimulate and really be
Mhm.
available to scientists for as long as not everything is destroyed.
Mm.
by
Uh,
analysis, so thousands
mhm.
of people.
Yeah. I also have a question. Because I know that there were some like political issues
Yeah.
and stuff, funding and whatever.
Oh,
Yeah,
still an issue.
yeah. And some plans are maybe postponed or whatever. So it's like touchy. It's a touchy subject, I
It's a
guess.
touchy subject. Yeah. When
Yeah.
I joined, the idea was that I think they would pick them up. The mission would be over by 2028, and the
Mm-hmm.
samples were expected by maybe 2030. Then it
Mm,
moved to 2031, and now it's not
yeah.
clear if and when it will
Yeah.
happen at
Yeah.
all.
Yeah. It's a yeah. I mean,
There
it's
was
it's
some
a it's
mission.
a yeah, it's a little bummer. I
It's a
guess
bummer, yeah.
so far.
But
If I were a Chinese president or something, I would say, oh, we're gonna make something. Go there. We don't wanna your sample. We're gonna take our sample at the same similar spot, and then we're gonna bring it back, and then we're gonna analyze it, and done deal. I mean, if I was a president of China, which I'm
China,
not. So
China indeed has, you said it, not I, but the sample
okay,
will be there on the surface of Mars for really, I don't know what space law dictates
yeah, yeah,
who can pick them up or not. But China does have a space program, and China is planning a mission to Mars to collect, they will just collect some soil. So
okay,
regolith from the surface of
yeah,
Mars and bring
mm-hmm.
it back by, I think they're launching in 2028, and that should be a
Yeah,
quick mission. So
yeah. So
actually planning something along the lines.
yeah. So I wouldn't be
I guess,
surprised. I wouldn't be just a couple of little questions. When that if that I'm gonna say when that when those samples arrive to Earth, what would be the main like the key analysis that we are expecting? Which results do we want to hear? What types of analysis?
certainly this sample 25,
Okay, yeah, of course,
should
yeah.
be uh, any sample where there's potential to perhaps find fossils of microbes or
Okay.
organic matter that should be analyzed. I think one of the major really questions that we still have
Hmm.
is how old are these sediments? How
Okay,
old
so
was,
that's yeah.
meaning when was there water on Mars, on early
Mm-hmm.
Mars?
Mm-hmm.
So finding minerals that we can date
Mm-hmm.
would be, so there are a lot of analysis. It's really, this
Yeah.
is the beauty of the sample collection. It's not
Mm,
one thing. It's really
mm,
a combination that really builds a very cohesive picture.
okay.
This is a
And
lot.
I
Of
think
course,
yeah.
25. For me, that would be looking and sectioning the leopard spots and looking
Mm-hmm.
for any distributions of organic matter, because we don't know
Mm-hmm.
where that organic
Mm-hmm.
matter is. Is it even in the leopard spots, or is it, who knows where it
Okay.
is? Because,
And I
yeah,
think you mentioned somewhere, and I think that there is in the paper that one of the words that were key for me, isotopes. So I guess that isotopes are really I mean isotope
yeah,
like analysis, right?
yeah,
Because it can be a telltale sign,
yes,
smoking gun, somebody said, right?
smoking gun.
Of
Yeah,
life. Life has a
yeah,
particular way uh, of like filtering or whatever, working with one type of isotope and faster with one type of isotope
exactly.
and yeah. So I guess when isotope analysis comes, I guess that can be really also very
Yeah,
cited and quoted
yeah.
paper,
Oh,
influential
yes.
paper or papers, right?
Oh, papers,
Yeah,
because you
yeah.
could look at those sample 25 has sulfate minerals,
Mm-hmm.
so the
Mm-hmm.
oxidized form of sulfur, and has sulfide minerals. And life has a very specific way of selecting for light
Mm-hmm.
isotopes of sulfur. So
Mm-hmm.
if we are talking about some biological process that reduced sulfate to sulfide to
Mm-hmm.
make
Mm-hmm.
regite,
Mm-hmm.
those
Yeah, yeah,
darker minerals,
yeah.
you could expect quite large fractionation, so large isotopic effects.
Mm-hmm. Yeah, because life
And
uses enzymes, life speeds up the
yeah,
reactions, and life
exactly.
needs lesser energies. And that's what related to that scenario of high temperatures, low temperatures. If you have low temperatures and enzymes, you can have these types of
Large
reactions too.
differences. Yeah.
Yeah, yeah,
And
yeah.
if it's almost very little difference, then, well, maybe we should consider some abiotic processes really more seriously.
Or some prebiotic, right? Prebiotic
There you go.
something.
There you
Yeah,
go.
yeah, 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 yeah.
Prebiotic.
Yeah. Which will be also very exciting, very, very hugely exciting. Okay, Tanya, I don't want to bother you too much more. I'm just gonna go to that last topic. Sometimes I forget to ask my guests what do they do when they don't think about like Mars and stuff and minerals and
Mars and minerals and
yeah,
microbes. I do
yeah.
I spend quite a bit of time doing that. And
Yeah.
when I don't, I like to read. I like
Oh,
to
cool.
travel.
Awesome.
Yeah. I like to, as I told you before, I read fiction a lot.
That's awesome.
I need some
What?
break from
Uh-huh.
science.
Cool.
So I do not read popular science.
Cool.
Yeah.
Totally legit. If you ask me. Okay.
Okay.
Did you read something really great in the last, I don't know, couple of months? Something that was like really awesome.
I just started. So I've only read one story, but
Okay.
this is science fiction.
Cool. Okay.
Cheng.
Oh, okay.
I
Yeah.
yeah. I
He's a
loved
famous
it.
guy.
He's
Cool.
a famous guy. Yeah.
Mm.
He's I think not contact. Sorry,
Uh-huh.
what am I saying? No,
Huh? Okay.
heresy.
No, it's
But,
cool.
what is the movie based on one of his? It was more recent, with
Um,
Amy Adams. It was about a communication when aliens made
Arrival.
ah, Arrival.
I just
Exactly.
did a quick Google search. No,
Yeah,
I
good,
didn't know
good.
that. I
I
thought
should.
which we
Yes,
watched.
right.
Okay. Mm-hmm.
Yeah, that's a beautiful movie, and so I'm yet to read the short story on which this movie was based, but I really enjoyed that. I'm reading multiple books at a time, so I'm
Cool. Of
reading
course,
something
everybody
that
does. Everybody does.
nonsense.
Uh,
Okay, makes me feel like guilty.
do
I'm
you?
sleeping.
Cool. Do you? I guess it was in English. So, but do you sometimes have a like an itch for reading something in Croatian or is it like totally irrelevant?
Oh, I love some Croatian authors, and I
Okay.
would really want if you have recommendations for Serbian ones.
Oh, okay. Okay.
Damir Karakaš,
Ah, cool. Yeah.
yeah,
Oh, he's hardcore.
and
Though.
it's
Yeah.
hardcore. It's generational. Then
Yeah.
Zoran Feric, I like a
Oh,
lot.
okay.
Yeah,
Okay. I my one of my favorite Croatian authors is Dežulović, of course. Dežulović and Tomić.
Julovic, of course. Yes,
Dežulović and Tomić. I don't know which one is the best.
and
When I
Tomic.
want
Yeah.
some happy thoughts, and then I read, of course, Tomić because he's able to write about awful things and to make me
and
laugh.
and be funny. Yeah,
Yeah, people are
yeah.
killing each other. Ha. Very funny. Uh,
You always find some angle.
yeah,
Yeah.
yeah. He's so great.
And
And also the
I like the guy from Split. He writes it's murder mysteries
okay.
in Split and the islands.
Mm.
Jurica
Oh,
Pavici.
okay. I think I've heard. Yeah.
Yeah, he's really good. I love murder mysteries
Oh.
mostly because they convey when they convey a sense of I don't care who's done it, but it's more about a sense of place, and it's always the socioeconomic and
Mm.
sociopolitical
Okay.
aspect of so,
Yeah,
and
yeah,
I
yeah,
think
yeah,
he
yeah.
does it well.
Well, weirdly enough, currently I'm reading a novel of Serbian by a Serbian author.
Okay,
Ivan Tokin, he is his
even
name.
talking.
Yeah,
Okay,
it's his newest book. I forgot how it's called because it's like a sentence, whole sentence, but it's about serial killer, a woman serial killer. So it's like
whoa,
a
okay,
thriller, and I really love the way he writes. I've loved the way he writes. I think I loved all his books. He wrote a couple of them.
okay.
So this one is
I'll
a
look
little
for
bit,
that.
yeah. This one is a little bit nihilistic. She kills
That's all right.
people. No
Keeping with the
emotions.
territory,
Yeah,
all of them are.
yeah. Which is weird because he also wrote a beautiful, beautiful book called Pas Dog. that's what I would recommend sometimes if you like dogs, or even if you don't like dogs. Pass. I don't know. It's like amazing, soft, emotionally beautiful, smart book.
Yeah.
But this one is also, this one about lady serial killer is also cool. I mean, he writes, he's very well at writing. I don't know if he had some experience or stuff, or he's just talented. But
He's just
I'm
talented.
enjoying.
I'll look it up. I
Cool.
took a note. I'll look it up.
Cool.
Yeah,
So,
that's great.
Tanja, is there something else you wanna say? I mean, we spent quite some time talking. I enjoyed.
Yeah.
We can switch to Croatian Serbian. Okay. Ja ću sada da zaustavim snimanje. Stop. I nisam ti se ni zahvalio. Sad ti se zahvalio, ne znam. Ali dobro, snimit ću kratki klipić, pa ću ti se zahvaliti. Ali svakako hvala ti puno, znači, što si posvetila vreme. To je to. Hvala puno. Važi, lep provod. Ćao.